So let us move to how this Cheminformatics is going to help in quantitative structure activity relationship. We know that so the QSAR is the evaluation of the molecular features that determine the biological activity. So when we change the molecular feature like a functional groups and its properties, electronic and steric factor functional groups, it changes the biological activity. How we are going to have that effect mathematically, we can get any equation in the QSAR. So the prediction of the compound potency as the function of the structural modification, we are able to know it. So this Cheminformatics is the one which we are going to use for this particular purpose. And we can use the same thing for the visual screening and compound filtering. Now, don't get confused with the visual screening. Actual screening we are doing in the laboratory, in the pharmacological lab, where we are going to find what is its analgesic property, antipyretic property, or its any effect which is having anti-allergic or wound healing or immuno modulation activity. These are all the screening techniques we are having in the actual laboratory, where we are using animal models or some methods where in vitro methods we are going to screen. But in the visual screening, it is a process of screening the large databases on the computer for the molecules having a desired properties and biological activity in the computer itself, virtually, not in the real world. So because of this, we can find new activity in QSAR. How the activity is getting changed, we are able to know by using the Cheminformatics data, where descriptors are used in the machine learning and they are relating to the biological activity. Now we are finding a new activity, whether it is present or not in a given chemical substance. So the major application of VS, what is this VS? Visual screening techniques is the identification of the novel active molecules in the large compound databases. So large compound databases, what we have already seen, which is created by the Cheminformatics. That one, all the chemicals which are already been kept 10 to the power of 60 odd, we can check it their activity virtually without entering into the actual lab using animal or touching a test tube. Now answer this question. The process of the screening large databases for the molecule with desired properties and biological activity. We are screening in the computer. You have to write that. Just answer the query. Kiran Teja answered already. You have to type. If you want, I will go back and see. Akshaya's. This question is for what you are going to put in that is not in the laboratory, in silico computers, what we call. 5 have already answered. Only 10 students have entered it. Now I will be again putting 25 students are there. Just they have to go and join by clicking this WhatsApp, whatever the link I have kept. They have to double click it so that they will enter into the background mode. Then they can answer the question to answer session. They can join. Tejashwi answered, Chandan answered, Sahitya answered, Shreya completed, Shruti, Sai Javali, Praveen, Pranita, Pranati, Dikshita Rao. So So make it complete fast. We will move on to next slide. Sir, can you please mute that music? Your voice is inaudible. Okay, I will mute the music because it is in the one what is making the noise. This music will appear only during QA because at that time I'm not supposed to speak. Okay, 11 all already over. The correct answer is VS, visual screening. Now attempt this question. Which of the following major applications of the visual screening? So 2 or more options are there. You can answer. Whatever the answer you feel, you write it. So we are going to use the visual screening for the designing the library of compounds which are having the novel activity and identification of the novel active molecules from the database of chemicals which are having very large number of molecules. Now the music will all automatically stops. Only it will appear in the assessment mode. Now we look into the visual screening. We have seen QSAR application seen with the Cheminformatics what we have gained. Because of that reason, we are comfortable now. We are using the computer system and computer aided drug design, computer assisted drug design. We are able to do. Another term which we can use is the computer augmented drug design. We are improving the activity by the augmentation because of the QSAR helped us. So this impact of the new technology and the drug discovery is, so it is revolutionary with respect to new technology which have already emerged. So one is gene chips and genomics, that is Human Genome Project, HGP. Bioinformatics, this is the next session we are going to have after Cheminformatics, and molecular biology. So it is started in the new phase one, Watson and Crick, they have identified the DNA structure and more protein structures which are not previously identified. Now it is revealed. High throughput screening, where we are using the laboratory robots and assaying the assessment virtually by using the robotic and automatic systems. We are evaluating fast in the laboratory in the HTS, that is high throughput screening, and visual screening and library design. What we have already seen, because of that visual screening, we are having a greater number of library of different categories of the chemicals which are having different biological activities. Combinatorial chemistry, where we are doing the multiple reactions at once and finding the activity, taking that particular mixture of compounds which is having more affinity or activity. And then we are going to select the best by the separation techniques, and we see what the compound is, and then we are going to analyze the drug likeliness. So other computational methods, these are all the advances which made us to get into this computer aided drug design, which is all based on this cheminformatics only. Now write this question and attempt it. You have to fill in the blank. Discuss how new technologies such as gene chips and high throughput screening and combinatorial chemistry have impacted the drug discovery. You can write as a sentence. So whatever you feel. Now I'm moving on to the so these technologies, what they have done. So they have reduced the time of inventing a new drug. They reduced the cost because their failures are the fast and early. And we have made the accuracy, a drug likeliness, we are able to find out near closer to the reality. And precisely we are able to design particular disease, drug for a particular disease. This is what we have attained because of these tools. So how the informatics can help out in this particular case, it is because it is producing the managing the information. Previously it is presenting as a data, it is now it is giving as a metric to reduce the risk by the virtual screening, library design and docking and cost benefit analysis. So we are able to get this particular task completed because of the cheminformatics. So it is making the information available at the right time and right place. It needs to be integrated into the process, which is already made by the computer and which we are previously doing manually. Now it is done by the computer. That's how it has helped us. So what are the softwares of relevance? I already told you each topic has got some databases, softwares, which you have already given as an assignment. So you are trying already and writing. If somebody has started it in the A4 size sheet papers, you write that also and start writing it. You complete it because this is one which we are giving in the click2drug.com. You can find it in advance. So the software relevance which bridge between the computation and the science is what the software which is having a goals, that is producing a compound that have the biological activity. That is actually what the science involves. In that also the software all present. And the task is work out the chemical synthesis and choose the good reagents and try. We are all entering based on the descriptor data. We can choose the correct agent and try the some documents which are having a reactions in the short path method. So shortcut method and bypasses so many number of trial and error method in the laboratory. That is science-based approach we have got some software relevance. You will find in that also in your assignment this type of science-based computation techniques. Again with Cheminformatics, we are going to have the software for the clustering and simulation, searching activity models, scaffold detection, docking, log P calculations. So these are all the things we can attain with the Cheminformatics. So this data can be further used for the further improvement. So what you can do? So the cluster analysis, we have got a similarity of the compounds which are having a similar bioisosterism or the lead, they can made as a one cluster. And we can identify the activity related fragments so that we can use those fragment in the fragment based drug design to get a new drug moiety, that is de novo drug design. So these are all made open, what we have learnt in the previous 3 sessions, which was possible only by this particular Cheminformatics and subsequent softwares what we have got. Now, which tasks are associated? See the question and attempt it. You can scroll down and remaining answer also you will find it. Is a single answer question. So Cheminformatics identify the activity related compound, that is actually the science which is choosing the good reagents so that we can use further. With respect to the science, it is going to help us, not the biological activity. Cheminformatics, where it has come from and where it is now and where it is going. That is the question in this particular is the past, present and future, what we say. Because of this advancement only, we are comfortable now. In the pharma field, we have got many drugs which are entering and we are having so many generations of the drugs. So from chemical information to Cheminformatics, we are able to integrate with the technology from the molecular modeling and developments in the computer hardware and software that is also resulted into a fast track, and data explosion arising from the development in the combinatorial chemistry and high throughput screening because of the robotics. And we are having the automatic systems. We have got so many inputs. Previously, we were not having. Now we are having the same all together. Because of that reason, the future is more promising, and we have very plenty of resources to invent a new drug because of these technological advancement. Now we look into that, what the advancements we perceived with respect to the time and cost, and these advancement with respect to the data and hardware and software, what we made as in a front foot to go ahead in the technological outbreak or the revolution, what we are perceiving. The first one is the molecular modeling. In that aspect, we have got too many advances, and the cheminformatics opened this field to the extent that we can find the reality in the computer without going to our crystallographic studies. Cryocrystallography, what we are seeing, such studies are not required to be taking place because the computer is doing the particular task. So the positioning of the putative ligand. So this is whatever the ligand what we are having, it is going to be promising into the protein active site. First attempted by the DOC program in 1982 itself, this molecular modeling, where we are going to make the drug likeliness with respect to the pocket of the protein. Initially restricted to the rigid ligands, because the technology is not that much is the flexible ligand we are not able to adapt, and flexibility making a ligand to flexible like molecule, it was difficult. And now it is possible for both the ligands and the rigid. We can go to the flexibility with respect to both proteins and ligands we can do now. The current program permits some degree of the flexibility. So using the structure-based design, move from the docking in a single ligand to a sequential docking of the large databases. And we are going to move from the docking to a single ligand to a sequential docking, means which we are having a multiple of chemical modifications. And we can create a large number of data sets so that we can have some lead or a pharmacophore. We can come out of pharmacophore with this particular task of a molecular modeling. So the lead, once it is there, lead identification is there, we can have a lead modification and lead validations. And further, we will make a drug likeliness scores, and we make a drug that is selected from the best score. So with the docking, we are able to do that. What the docking was to place the drug molecule in the pocket. So this is how the computer has seen the active sites and predicting the ligand fitting in the molecular docking, so that we are going to find these drug likeliness scores are very high or less, so that we can come out of a new drug. So what we have seen in the previous picture, we are able to look into the scores with the docking, so that we are able to see the fit is perfect or not. What we have seen the previous slide, what the picture is there, where we are able to visualization. That is actually called as a visualization of a ligand in a receptor protein receptor complex. So one more thing which we can create with this particular, not only the visualization with this Cheminformatics data and using the software, we can have the graph theory also. What is this graph theory? Graph theory is a branch of mathematics that consider the set of objects like a nodes, and the relationships are called as edges between the pair of these objects. Previously, you remember, we have got one type of thing is that we have got putting the proton donor groups, proton acceptor groups, and we have created the distance between them, and we have created the triangles, and we have selected one probatom, and these nodes are nothing but those proton donors and acceptors. And these nodes which will be having edges, like there when you are having a three-dimensional array, their distances will be different. So we take it as an edge, that is one between the pair of these objects, like we are taking from N group to, that is nitrogen group to oxygen. What was the distance when we are making a rotation of a flexible ligand in certain angle? So we are able to make that, and with respect to the probatom, we are making a changes in the rotation of the ligand in the pocket. We are able to identify which combination or what is the isostere which is having or isomer which is having a perfect fit, whether it is a cis isomer, trans, or the dextro or levo territory. So these are all able to know it by these node and edges concept. This is nothing but the graph theory. So this is, we have already seen, because of that reason, we have got free Wilson analysis, Hans analysis we have done. So these are all able to get the moiety which is going to fit in the nearby future without any problem. So we are able to detect it. So the definition of completely general, allowing the graphs to be used in many different application domains, as long as the appropriate representation are derived. So what we are seeing with respect to the R, so that R is acting as a one tool to us between the nodes. That R, that is the distance, will be keep on changing with respect to the flexibility of the moiety. This is acting as a great representation to us to come out of a better structural characteristics with a particular having a functional group that is going to give a better result as a drug likeliness. This is what the graph theory is. So now we will be moving into the next slide. What the cheminformatics is, the data has been converted into a knowledge. Now the question comes, how this has happened with the computer? Why, how it is able to do it? Because computer cannot visualize like a human eye, which is having a up and down, or it is having a steep, or it is like a burrow, or it is having a bent or a turns. So this is not possible. So it will be making use of a graph theory, where the nodes which are having actually a donor or acceptor, proton donor or acceptor, a particular property, that we are converting into edges, the distances, and we are looking into a mathematical graph. That graph can be easily studied with a 3-dimension or a 2-dimensional according to the property. So the computer is converting our data into its own understanding capability, and it is making use by itself by seeing that particular data as a nodes and graphs and giving as a knowledge as a output as converting as a knowledge. That knowledge will be lead to a action. This is what happened with the present with the cheminformatics, so that we are able to enter into the computer mode instead of the manual mode. This is what the cheminformatics is. Now you will be having a question, sir, how the computer is doing that particular activity, so that we will be making use of that particular one, and how the step-by-step procedure in the cheminformatics. So this is actually released by the PubMed as some guideline, and we will like to see that one also. Now we will introduce.